Hereby I open this academic ceremony at Maastricht University. First of all, I'd like to welcome Mr. Asim Cengiz Akbulut, and he will defend his thesis for a double degree of Maastricht University and RWTH Aachen University. And his thesis is called Spatiotemporal Developmental Regulation of Arteriosclerosis. I welcome all the members of the degree committee, in particular the three supervisors sitting opposite to me, and these are Professor Leon Schurgers. He is Professor of Biochemistry of Vascular Calcification at Maastricht University. Professor Rafael Kraman, he is Professor in Experimental Medicine and Systems Biology at RWTH Aachen University, and Professor Sanya Sinha, Cardiovascular Regenerative Medicine Professor at the University of Cambridge in the United Kingdom. I will introduce the 6 opponents later during the ceremony, because if I mention all these names, no one will remember that. Welcome to the audience here in the aula and all the followers of the live stream. Mr. Cengiz Akbulut, may I ask you to give a summary of your thesis? Please go ahead. And I wish you success for the coming hour.
Thank you. Highly esteemed pro-rector, members of the Corona, friends and family, ladies and gentlemen, and to all of you out there in YouTube land, thank you very much for being present here today as I defend the work of my doctoral thesis. For today's story, we need to go back to 1966, when Percy Sledge released his hit record, When a Man Loves a Woman. Now, this song might bring back fond memories, possibly after a candlelit dinner, holding someone special close as you slow dance to this rhythm and blues classic. But I would like to extend from the title of this talk to discuss what happens when a man loves a woman, when a woman loves a man, as well as in other circumstances, the consequence of which allows a cell to form. Then from here, a beautiful event of nature occurs. 1 cell becomes 2, 2 become 4, and before not long, 4 have become many more. However, at one point in this process, the cell needs to decide what it will be. Roughly based on where and when it happens to be, allows it to become the different parts of the human body. Once we are born, some of these processes continue and we develop into who we are today. However, so some of these decisions are governed by biology, but depending on how much you like Percy Sledge, it's likely a lifestyle choice that's influenced by culture. Some 10 years before Percy Sledge released this hit classic, Elvis Presley released his first ever studio album, the self-titled Elvis Presley. In the same year, Conrad Waddington proposed to humankind that within the concept of human biology, within developmental biology, so going from 1 cell to a living organism, that this should be considered that of a ball rolling down the hill. That as this ball rolls down the hill, it may enter crevices or valleys that once entered into, it may not roll back on. That these crevices and valleys are decisions that allow the different parts of the human body to form. In the first decade of the 21st century, scientists in Japan discovered part of this developmental process can be reversed in the laboratory. Roughly speaking, by using 4 proteins found commonly when life is no more than a few cells, we can use these 4 proteins to genetically engineer an adult cell to that of an embryonic-like cell, effectively rolling the ball back up the hill. These cells are commonly referred to as human-induced pluripotent stem cells. On pages 37 to 39 of my thesis, I show that from taking the blood from an individual, by using the proper laboratory techniques, that I could reapply this process, turning these cells into induced pluripotent stem cells. So from blood, I genetically engineer the cells to go back up the hill. We know that I've done this by checking whether or not they can be identified as stem cells, as well as seeing if they can behave as stem cells. And here what we show is that using these generated stem cells, we can make neurons, gastrointestinal cells, as well as muscle cells. Stem cell technology has great promise for developing new therapeutics that will better humankind. However, the word stem cell on its own, from a technical point of view, doesn't mean much. Stem cell is an umbrella term for a variety of different types of cells, which I like to categorize into 3. Firstly, we have adult or somatic stem cells, which are in all of our bodies, helping us stay alive. We have embryonic stem cells, such as those that are found in early development. And we have genetically engineered ones, induced pluripotent stem cells, such as the ones I just mentioned. There would be a great benefit to humankind if we could use induced pluripotent stem cells to generate cells or tissues used in transplants today. On page 41, we show generation of mesenchymal stem cells, a stem cell commonly found in our bodies, and we show that these cells can be used to make bone, we show that these stem cells can be used to make bone, cartilage, fat, and vascular cells. While we can make these different cell types, the various methods to make them are imperfect. What we make in the laboratory is not yet identical to that of what's found in the human body. From pages 41 to 47, myself and colleagues performed a series of experiments into questioning whether by introducing a single factor, that of vitamin K2, specifically menaquinone 7, could improve bone formation in our laboratory study. We found that menaquinone 7 in our experiments improved the bone formation by having an antioxidative effect. It increased the ability of these cells to reproduce, so replicate, and as well as to migrate, as well as increased the formation of collagen fibers. This was in addition to subtle changes to processes within the cell that indicate a more mature phenotype. Those of us who have aged are more likely to enjoy Percy Sledge as well as possibly other rhythm and blues classics. But also as we age, our bones become weaker. The paper that came from this research is part of a new generation of biochemical-based evidence that suggests vitamin K2 might improve bone strength and fracture healing. But I wish to put this disclaimer that you should always consult your general practitioner or other specialists you're seeing before taking supplementation, especially if you are on prescription medication. So far, we have established that as we age, we are more likely to appreciate rhythm and blues music, in addition to some biotechnological, almost innovation that can possibly make our bones get harder. But in life, something else can get harder that's not necessarily to do with Percy Sledge. And what I'm talking about, in fact, is that of our blood vessels. As a matter of fact, age-associated hardening of blood vessels, commonly referred to as vascular calcification, is highly associated with that of bone loss and increased fracture rate, particularly in postmenopausal women. With this in mind, we wish to use induced pluripotent stem cells to make blood vascular cells to investigate whether we can use these cells to study vascular calcification, the deposition of bone-like material into our blood vessels that causes them to stiffen. From a developmental point of view, one thing which I find fascinating about the vascular system is that while we can say that the brain forms from a particular valley and deliver through another, the vascular system is composed of multiple valleys which align up at the bottom of the hill to give us our cardiovascular system. Some 10 years ago, in the laboratory of my co-supervisor, Professor Sanjay Sinha, they discovered that, in fact, we can use the developmental potential of pluripotent stem cells to make different vascular cells. On pages 47 to 48, I showed that I was able to successfully reapply these processes and generate different types of vascular cells. Considering that vascular calcification is commonly referred to as an age-associated disease, and pluripotent stem cells are considered by many to be immature, we questioned whether these cells might even be capable of calcifying. In pages 48 to 49, that we see indeed these cells are capable of calcification. Next, we questioned whether or not there might be a difference between the particular vascular cells. In doing so, we identified that one particular population of vascular cell, that of the lateral mesoderm or cardiac smooth muscle, in fact calcify more and faster than the other populations. We wondered why might this be the case, and so we had a look at what's happening inside the cells to see what the differences might be. We expected all different populations to undergo a variety of changes under this calcification disease modeling. But to our surprise, we found that the population which calcify more and faster didn't undergo any changes, or any significant changes, whereas the other two populations did undergo changes. When we looked at what the differences are between these populations, we see that there is it leaves us with the question as to whether or not there is a developmental priming of our body that allows this particular cardiovascular disease to develop how it does. Further from this, We hypothesize that if there's a developmental priming, that this is something which has likely been driven by evolution. But now I know, looking at the faces of you all in the audience, some of you are wondering, so what does this have to do with Percy Sledge? Well, let's look at the bigger picture. Percy Sledge didn't come out of nowhere to create a love song that would live on through the generations and be talked about here in Maastricht some 60 years later. There is an origin about it. Rhythm and blues was not invented by Percy Sledge. Going one generation of music back from Percy Sledge, we can go to the swing era, a swing era which was built out of, which came out of the Harlem Renaissance in the 1920s, which rose from the birth of jazz and blues music cultures which are built upon African roots that were maintained by formerly enslaved peoples. So there is an origin that can be traced. Much like there is an evolution of music, life itself has its own origins. A centerpiece to our existence was when life came out of sea to the land. On pages 55 to 57, I discussed the evolutionary origin of a protein considered by many to be the most potent inhibitor of vascular calcification, that of the matrix scar protein. By looking at the chemical composition of this protein, we see that it varies between species, but in fact we conclude that once life stepped onto land, differences to this protein became much less. Further, we found out that this protein is not expressed when life is but a few cells. Life needs to become more cells and already some way down the hill before the matrix scar protein starts working. So we also identified roughly at what point after listening to Percy Sledge does this protein become active. We also understand better how it's expressed in the heart. At this point in time, excuse me, at this point in time, I wish to conclude the presentation of the findings of my thesis. But before I open the floor to questioning from my highly esteemed opposition, I wish to ask you the question as to why are we here? Maybe it's because I or my supervisory team invited you to be here. If, like me, you have immigration in your familial background, then in some ways you are here in part due to whatever reason someone decided to leave their homeland. But at the same time, if you are here for this defense, then in some way the reason why you are here is because someone in my family decided to leave their homeland. If they didn't make that journey, where would you be right now? The world we live in is a beautiful thing. Since a young age, to look at the diversity of life and culture on this planet has always struck me with amazement. It is with great wonder that I have always asked, why is it like that? On page 71 of my thesis, I present the idea that at the center of our existence is that of biochemistry. Internally, our biochemistry is regulated by genetics of a cell, cells that work together in a system which make organs. These work internally within a system of the human being, and we are all part of a network that in a larger sense is governed by the rules of the universe. I believe that nothing happens in isolation, and there is always a reason as to why things happen how they do, and cardiovascular disease is not exempt from this. On page 79, I conclude with how research presented within this thesis fits into this conceptualization, that aspects presented within this thesis are not static but self-influencing. Further, that life itself is a self-influencing entity, and we are all intertwined, and whatever we do has an impact, no matter how big or small we think it may be. Lastly, I wonder, where would we be, or more so this presentation, if it wasn't for Percy Sledge? I would like to thank you very much for your attention and for listening, and with this, I would like to formally end my presentation and hand the word back to the highly esteemed Pro-Rector.
Thank you very much for your clear, philosophical, and nicely illustrated presentation. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Tilman Hekking. He is Professor of Biochemistry at Maastricht University, and he is also Secretary of this committee. Professor Hekking.
Thank you, Mr. Pro-Rector. Dear candidate, I need to call you dear candidate because I'm not allowed to call you young Mr. Akbulut or grasshopper as you usually do, but dear candidate. First I would like to compliment you with this beautiful thesis. It's a fine monography based on a set of really fine papers, and of course I would like to include your promotion team in these compliments. But then again, we need to also discuss about content. But not before I said that before you made your very wise decision to spend the rest of your life with us in the Department of Biochemistry, we were in the dark. And I would like to really thank you, and also from our institute, that you brought IPC technology to us, because I'm not sure that everybody knows that without any help or guidance of your scientific parents, together with your colleague Roger Veldrop, by yourself you set up this whole IPC technology in our institute, leading to this beautiful stem cell lab now, of which you are the youngest director ever within KRIM. So congratulations on that. But now about content. Let's go to page 11, where this childhood drawing of yours depicts the structure of the aorta. And as you already said in your presentation, you can see that there's different embryonic origins of the different parts of the aorta, and especially the aortic root, which is very important here, the arch and the descending aorta coming from the lateral mesoderm, the neural crest, and the paraxial mesoderm. When you then look, which you also showed in your introduction, on page 50, and you look at the calcification propensity of these lineages, then you see that this lateral mesoderm, actually representing the aortic root, sort of, yeah, if I can say that, heavily calcified. You have maybe some explanations for that, but what is the meaning of it? Do you think that there should be a reason that the aortic root should be a little bit more stiffer?
Highly esteemed opponent. Thank you very much for the compliments you made, as well as for being here today. Further, I would like to say thank you for heading the department, which I was fortunate enough to spend 5 years wherein I conducted this PhD work. It's a great pleasure to have you on the board to question and provide questions for today. With regards to the lateral mesoderm propensity for developing calcification quicker than the other populations, partly I believe that this is due to evolutionary conservation of vascular development versus that of bone development. And so I think that while from a biological point of view, people isolate, pick a favorite cell or pick a favorite organ and study that. Actually, if we look at the genes which are necessary for forming the cardiac mesoderm, there's a lot of overlap with that which is required for skeletal bone formation. And so that's why I think there really is this developmental priming which has arisen as to why these cells calcify faster than they do.
It's not on purpose. It's not in design.
I think if we were to say that it shouldn't be like this, it's an accident of nature.
Okay, that's cool. Yeah, thank you. If you then consider one of your favorite proteins, and starting to become also one of my favorite proteins, the great protector of systemic calcification, MGP, you already talked about this. And when you then look at page 58, you say that there's a greater expression of MGP at the ascending aorta and valves. So this sounds to me a little bit counterintuitive. There's like a lot of MGP, but there's also a lot of calcification. So how is that possible?
So how I would explain how it's presented within this thesis, within this book, is that where I'm referring to the high expression of MGP, that this is something which is potentially a developmental regulation, where there's a role of MGP that's developmental. Furthermore, for this to maybe be coupled as to why calcification typically presents itself at the valves or at the root, is that maybe I'm still investigating this, but should this higher level of MGP being present, then on loss of MGP or inhibition of MGP, potentially by medications such as vitamin K antagonism, such as warfarin, would remove this calcification protective effect that MGP has, which is maybe why it's there and needs to be there.
Yeah, this is a very nice explanation, but still, there is MGP in a relatively high level, and it cannot prevent this calcification.
Yeah. So at one point in our life, by kind of how can I express myself how I would like to?
Yeah, it's very difficult because it's very counterintuitive. This is what I said. So
yeah. So it's a protective mechanism that is there, but by just one example of having, let's say, a lifestyle which is not encouraged by medical authorities, by creating damage and disrupting the homeostasis by which our body is in, that this causes that during ordinary cellular turnover and during ordinary mechanistic processes, at one point the body becomes unable to keep up with this.
Right. Your mentor always tells me that there's calcification, so there's more MGP being produced and expressed to overcome this calcification. Could you live with that?
Yeah, so I think definitely there's this feedback, but also with kind of where the mechanistic appreciation in our laboratory is going towards on MGP, is that potentially you have this almost like inconsequential feedback of that by having a calcification present within the valve or within the aortic wall, you then are putting more stress and strain on these on the smooth muscle cells, inducing more of a contractile phenotype, and then as a compensatory mechanism, an almost synthetic phenotype wishes to be induced by it in the regulatory properties of the cell, which increases the MGP expression.
Okay, I can live with that. I have is there a short time for questions? So if you look at the MGP sequence on page 57, you can clearly see that the first 9 residues are tyrosine, gamma-carboxyglutamic acid, serine, histidine, glutamic acid, serine, and then finally methionine and glutamic acid and the serine. And all these 3 serines can be phosphorylated, and they are phosphorylated. And one of your colleagues, Stijn Achten, showed that they aid in inhibiting calcification. And so for me that's actually kind of unique. So here the phosphorylation machinery is in the cell, MGP is mostly acting, I think, outside the cell. So this will be a first like phosphorylation activity functional outside the cell. So is this like coincidence because of the charge of the phosphor groups, or is it true? Do you think that phosphorylation has a extracellular role?
You may give a short answer, please.
Possibly.
What?
Possibly.
Possibly. That's as short as you can get. Well, I'm very happy with your answers, and I give the word back to the corrector.
Thank you, Professor Hacking. The opposition will be continued by Professor Andrew Baker. He is Professor of Vascular Biology at Maastricht University, but also at the University of Edinburgh in the United Kingdom. Professor Baker.
Thank you very much, Prorector. Dear candidate, congratulations on the thesis and a very nice presentation, and to the supervisory team for their help in this. It's a nice piece of work. Could I just ask you to read out proposition number 4, please?
Would you like me to read it, or
yes,
one
please.
of my paranymphs?
One of your parents.
The molecular and developmental environment that we as humans find ourselves in may direct pathological mechanisms and is a consequence to our evolution as human beings.
Thank you very much. I'd like a little bit of context on this. So can you maybe give us a little bit of information of what that really means in an adult in terms of perhaps genetic predisposition to disease, but more commonly, what does that mean for the different types of smooth muscle cells and how they may respond pathologically to injury of the vasculature?
Yeah, I'd love to. Okay, highly esteemed opponent, thank you very much for the question that I would like to jump into to directly answering. So with regards to let's say the molecular and developmental environment that's evolutionary driven, this is where I think one thing which comes to light as to really with a critical turning point for vertebrate for the formation of vertebrates was that of the influx of calcium carbonate into the ocean, which allowed for which altered the biochemistry of early life and allowed for skeletal formation to occur. And by having that kind of this evolutionary event and the allowance of a formation of skeletal structure, then this allows for vascular calcification as a disease to also be present and affect us in our lives. If you would like, we can discuss later about the genetic aspects of this.
But I mean, what I'm getting at is the different types of smooth muscle cells and the sort of perceived heterogeneity of smooth muscle cells in the vessel wall. I mean, there's been quite a lot of work recently from many different groups about how that plays out and how that may respond to injuries such as clonal expansion of individual populations. So how does the work that you've shown developmentally impact on that in maybe the adult system where cardiovascular injury happens over time?
Thank you very much for that question and for clarifying where you would like to discuss further. So from this is why I'm really appreciative for having the techniques which were developed in the University of Cambridge 10 years ago, which allows us to really have these isolated smooth muscle cell subpopulations, and so that we can really ask this question. Because if it wasn't for this technique, a majority of studies which we refer to use animal models, which they have their drawbacks, which we could discuss at another point. But as well as when you look at the human studies as well, we have this inability to really trace back as to what the lineage of this cell is. You cannot create a lineage trace human. So there's really this kind of open question where the development of these protocols have really allowed this kind of question to be asked, and where this thesis comes in as being one of the first times when we're able to ask this question in the context of calcification.
Okay, and within these different populations of smooth muscle cells that are in the aorta, and then you recapitulate with your IPS model, within them, are they all the same, smooth muscle cells, or are they within them they're heterogeneous? I'm not clear on that. Can you just clarify that?
No, thank you. That's a really good question. And for me to answer, I'm afraid I need to refer to new data which I haven't included in this thesis. So we've now recently performed single-cell sequencing on the lateral mesoderm smooth muscle because it brought to attention our interest. And from allowing Surat to do just the clustering how it does, my colleague in the department in biochemistry and cardiology, Deepak, identified that in fact there are 10 different clusters within this when you ask Surat to perform its analysis and tell us what we have within it, tell us what's in it. But actually how it presents itself from the Surat is that it's a clonal population itself. So actually I'm leaning towards the side that this is a very relevant model that can be further exploited in order to better understand the human condition. Okay,
thanks very much. And then sort of pushing on a little bit in terms of, this is a complex environment developmentally, and then in the human, and also in animal models, of course, but you're reliant also on in vitro models that you describe. Can you just, people obviously use these for disease modeling well, but what are the limitations of that as a sort of a much more simplified environment for studying this sort of problem?
I could probably stay here until tomorrow to discuss the limitations behind in vitro.
Give me the key points. Give me the top three.
So the top three which come to mind are that of we're using fetal calf serum. That is, I think, one of the number one things which is to be addressed, but it's anyway a general drawback of in vitro cell culture. Within the human body, we have one culture medium, and that is whole blood. But as humans, we are unable to culture cells as far as I'm aware, in really any way, shape, or form using whole blood in vitro, and I think that's one of the biggest drawbacks to using cell culture models.
Okay, and is there any way around that by creating more complexity in the model systems, or is where is the field going to go? What's your vision for?
Yeah, so what we've published, which is recognized in the publications within this thesis, is a work which I supported, where we used instead of fetal calf serum, a human serum from different individuals as well as patient cohorts, and we see that we have a differential response in this disease model based on that serum, based on using patient serum. It's still used in the same proportions as you would use fetal calf serum. So it's not a direct replacement for whole blood, but this is, I think, a very interesting avenue which we're further looking into.
Okay, and one more question, if I may. In terms of the differentiation model and the sort of sequencing analysis you did with the deep sequencing, there were no differences, if I remember, between the lateral mesoderm and the calcification. I find that very surprising. What is your explanation for that? And is that based on how you set the criteria for looking for differences between the two populations?
Yeah, so with regards to purely the assay base, so one thing which I control for, which I think is very important to control for in this in vitro kind of experimentation, is that you really control for passage of your smooth muscle, even if they are generated from different, from IPS, as well as to seed them all onto the same plate, so that if you perform a real-time imaging, such as we do with the red fluorescent probe, that you don't introduce plate-to-plate variability.
Okay, all right. Thank you very much. I'll hand back to the Pro-Rector. Thank you.
Thank you, Professor Baker. The opposition will be continued by Professor Rebecca Schneider. She is Professor of Cell and Tumor Biology at the RWTH Aachen University. Professor Schneider.
Thank you very much, Pro-Rector. Dear candidate, I would also like to take the opportunity to first congratulate you on this beautiful piece of work. It was a pleasure to read. And I also have to say I really enjoyed your presentation, very clear and very nice illustration. And I, of course, also would like to congratulate your supervisors for this beautiful work. With that, I would like to get started with a more kind of like open question. So, I mean, you beautifully showed, and I mean, as we heard, that's a technique that you also developed to generate iPS cells. And then you show that you can differentiate them into mesenchymal stromal cells, so iMSCs. So how do you kind of like see these MSCs? How do they compare to an in vivo situation in the body? And I mean, you also applied single cell technology, so we learn more about stromal subsets. Do you have any idea where they are exactly? Is it really MSC? Are they somewhere in fibroblasts? What's your opinion there?
Yeah. Highly esteemed opponent, thank you very much for your compliments and for being here today. It's greatly appreciated to have you in Maastricht. If I may jump straight into the question. So with regards to this model wherein I present the mesenchymal stem cell, I think I would like to take this opportunity to be critical of my own work and to say that this isn't this model could be better. There is a next generation which can be improved on it. I think it's a very reductionist-based system. Further, with regards to mesenchymal stromal cells, I think this is a fascinating emerging field. It's not one which I'm really strong in, but I really believe that the developmental heterogeneity, and possibly not even developmental heterogeneity, but when looking within different tissues, even just the spatial placement of these mesenchymal stromal cells is quite a critical component of their kind of profile, if you will, and sort of like how we can maybe decide that this mesenchymal stromal cell is different from another. But if I may add as well, I do actually wonder as to what the nature of this plasticity is, and I kind of have a feeling like from how I've been getting into my research with the smooth muscle and where the overlap of stromal cell is, part of me wonders if within the human system we have maybe a hierarchy of mesenchymal stromal cells, wherein maybe more a particular stromal cell is more plastic than another. Yeah.
So would you have any idea where your kind of like generated cells are positioned? Do you think they're more downstream differentiated or would be more up, kind of like, I mean, in particular, when we look at other cell types and you differentiate them from iPSC, they're often more kind of like in a developmental or kind of like early stage. So where would you see this cell type?
Yeah, so the mesenchymal stromal cell which I generated and we performed experimentation on, I consider it to be generic. It doesn't have a particular developmental induction. In order to generate it, it's purely take a pluripotent stem cell and apply serum with FBS on it and allow it to overgrow. So you have this really random differentiation process that goes on. It maintains a degree of plasticity, but I think it's quite generic. And I wouldn't, I think I have, it's not included in the thesis, but I have applied the lineage specifications and then switched into mesenchymal stromal differentiation, and that has produced a positive outcome as to a cell which has this mesenchymal-like plasticity. So I think that would be kind of like where potentially I could use these skills in order to generate new subpopulations of mesenchymal stromal cells.
Right. Yeah. So would you think there is a certain? I mean, you start from blood, which is, I mean, it's a good source. I mean, it's the easiest source then to get to do patient-derived iPS cells as well. But do you think there's a certain kind of like somatic memory? I mean, years ago was shown that if you fuse, for example, an embryonic stem cell with a somatic cell, there's some kind of memory. Does that exist for iPSCs as well?
I think most definitely it does. That not only do you whereas by inducing this pluripotent state, you do have partial reset of let's say epigenetic landscape. But for example, one thing which to be aware of when using blood as the source, which probably applies for any cell type, is the nature of somatic inductions within cellular processes. And blood is one of the organs which is really pioneering this line of research. So I think that's like a next level experimentation in order to really have an appreciation of the somatic mutations induced to PBMC-based populations, and then what happens to the iPS thereafter.
Yes,
but
that's
many
a whole
work
line
to do.
of new work. Was also more an open question. question. But so going into the differentiation, so when you basically add vitamin K, you show that you improve osteogenic differentiation. Do you think, I mean, I would have thought this would be counterintuitive. You would think maybe more adipogenic, so you have like this more, I mean, it's a multipotent state probably, but then you prime to a certain differentiation. Is that something that you expected before? And do you think it's really specific to the osteogenic, or does vitamin K anyhow promote differentiation of cells or keep them more?
Okay, so with regards to the model, so we always made sure to include a vitamin K control on its own. So I think part of the belief behind the hypothesis is that it's adding vitamin K within this developmental process. Sorry, and what was the
So do you think, because for example, I mean, you would think it, I mean, in a body, in a patient with cardiovascular disease, you would give it to prevent calcification. If you go to, I mean, we are doing work together on bone marrow fibrosis, there you have like, also new formation of bone, and you would think it prevents it, but rather if you have it in vitro, it promotes osteogenic differentiation. So.
Thank you. My apologies for asking you to repeat yourself. Memories triggered back. So by having this control of MK7 alone, it's really the MK7 within this forced bone formation, and this is, you know, an in vitro reductionist model. It's almost, I'm still unsure as to how relevant it is as a model when you have 10 millimolar phosphate with this high levels of ascorbic acid and dexamethasone. So it's just kind of like adding on to this already like reduced model, and it's building a model on a model.
Yeah. Do you have time for tiny? Okay, so if you look at your single cell sequencing, the MGP is quite broadly expressed, right?
Yes, in the developing cardiovascular system.
Yes, exactly. Was that what you expected? Kind of like based on also kind of protein level, I mean, stainings that you did and so on?
To be honest, I was very excited when I came across this heart atlas, and I didn't know what to expect. It kind of leaves more questions by being so widely expressed. So yeah, that's kind of where it's left us, which is why I presented it how I did, with looking at like the canonical markers of atherosclerosis to see kind of where this hypothesis, which is coming out of particular groups in the US of development and degeneration, are really coupled together. So I would have expected actually the single cell to. So regardless of how wide expressed MGP is, I would have also expected genes such as biglycan or decorin to really be coupled with it, and so that was the real surprise which came across when looking at this data set.
Very interesting. Thank you very much.
Thank you.
Thank you, Professor Schneider. The opposition will be continued by Professor Catherine Shanahan. She is Professor of Cellular Signaling at King's College London, UK.
Thank you, esteemed Pro-Rector. Yes, dear candidate, great thesis, enjoyed reading it, well done, very nice presentation. Lots of philosophical things in this thesis that are really difficult to ask in such a short period of time. So I'm going to leave that, but I think there's hours of conversation to be had around some of the things that you've presented to us. I want to get to the nitty-gritty a little bit about some of your studies of calcification. So you've got these three lineages that calcify at different rates, and we know that there's lots of mechanisms of calcification. So in your system, what was the mechanism of calcification?
Highly esteemed opponent, thank you very much for coming here. I really appreciate you making your way to Maastricht, as well as having someone from my hometown of London being here asking questions. With regards to the calcification, so what we hypothesize within our laboratory is that by using calcium alone, we have this quasi-intimal calcification induction.
So what does that mean? Like, what is happening to the cells that they're calcifying?
That by exposure to purely elevated calcium level, that this is the effector on the cells that will trigger the calcification.
But, like, you know, we know that maybe sometimes alkaline phosphatase is involved, sometimes apoptosis is involved, sometimes RUNX2 osteogenic differentiation is involved. what caused the calcification here?
In all populations or in?
In however you know. I mean, you've got data on RNA-seq where you saw 2 of the populations changed, but you didn't give any data on what changed in those populations. So what is the mechanism of calcification? Because surely that is going to be the question that's going to tell you why these LM cells are different.
Okay, thank you very much. I must start off with saying that when this RNA sequencing data came in, there's really a lot of avenues in which we can begin to explore the analysis. And so my apologies for not including in hundreds of different tables on differentially expressed genes. I decided against it. One thing which came from this analysis, which was actually momentarily disappointing, if you will, is that there wasn't an easy answer to give you. That there wasn't a single category of differentially expressed genes across all populations. I had this moment of maybe naive hope that, I'm going to, maybe we're really going to find like the key of calcification. But calcification, as many of us appreciate, is a far more complex process. And this developmental heterogeneity is one thing which we need to consider when looking at our cell-based systems.
Yeah, I mean, interesting answer, but if I was going to look at a gene set, knowing all of what we know about calcification, the first thing I'd look for would be osteogenic markers, because that's what billions of papers say is driving calcification. So if they weren't there, then I would think, okay, I'm looking at some other system here.
Right.
So what could the other possible inducers of calcification be? Because you haven't really looked at anything. You've looked at RFP for fetuin, binds to hydroxyapatite, could be calciprotein particles. So I just want to get you to think about what you think happened in this dish.
Quite. Yeah. So I think one thing which we found from within this dish is that with particular subpopulations of the smooth muscle cell, this transdifferentiation to a calcified phenotype needs to occur. And that's really kind of, I think, the takeaway message from the calcification, from the RNA sequencing data in that respect. But then when it comes to for this model of aortic root calcification, or possibly even valvular calcification, then really there is this kind of already priming, and that these cells will readily just take, if there's elevated calcium and they need to produce matrix, they will happily take it and put it into their cell wall. Whereas the other cells are more programmed to kind of think, I don't necessarily want to take this calcium and put it into my cell wall, and they need to be forced to change their mind, if you will, change their mentality, which is the transdifferentiation, in order to have that. And so to take a step back then and look at the differences between, like as I present on I think one of the following pages, is that as is when looking at page 55, the table wherein looking at the LM population, that regardless of calcification, these cells already have a baseline higher expression of many of our canonical markers of calcification. And if I may add as well, so MGP was not differentially expressed across all 3 populations. It was quite equally expressed. But then with regards to the differential expression at the neural crest smooth muscle cell, you had a significant increase in MGP in the neural crest population.
But I guess, I could pose another question and say, well, another mechanism of calcification is exosome release. Did you test whether they had more exosomes? Another mechanism of calcification is apoptosis. Did you measure whether there was more apoptosis? So how are you going to dig down into this mechanism?
Yeah, so that's what I'm doing right now, if you will. I think within the lateral, and this is where I'm still questioning kind of like the scientific, like my scientific conduct on this, as to knowing what I know as presented in the thesis. What are the next steps? Do I look to tackle all 3 populations? Do I look to dive into one subpopulation? Within the single cell of the lateral mesoderm, which again, my apologies, is not presented within the thesis, but would happily send to you and discuss later, there seems to be, there is one of the 10 clusters which is present is that of an already quite dead population. And so then we can think that, okay, so if this cell isn't undergoing a transdifferentiation, but then there's quite a relatively large number of dead cells within there. So possibly apoptosis is the driver of calcification within this lateral mesoderm population.
exactly?
Food for thought, I think.
Okay,
Yeah.
one very quick question. You used one cell line to produce 3 different ones. You do it 3 times. Are they biological or technical replicates? And what would happen if you used a different line?
Yeah, that's a great question. Great question. I think that they are biological replicates because they're made at different points in time. So it's not the same as taking from one plate and then putting out 3 different samples and then saying, okay, I have my 3 technical replicates. It was really, you have your IPS, it's the one singular line, and then months apart, these were made. So I think the genome is controlled. I could understand why one might argue that these are technical replicates, but I think that that these are biological replicates. And if I may just add, we did this because I wasn't too sure about the variability. Now that I have this data how I do, I'm very interested in getting 2 or 3 more lines, just making one, and then putting it in there and seeing where it fits into the clustering.
Yep, I think they're technical replicates.
Thank you, Professor Shanahan. The next opponent is Professor Christopher Hughes. He is Professor of Vascular Biology and Vascular Biomedical Engineering at the University of California at Irvine, the USA. Professor Hughes.
Thank you. And yes, may I offer my congratulations to you and to your mentors as well. It's a tremendous amount of work and some very provocative findings, I think, and an excellent read as well, as many have pointed out. So I have I guess a combination of technical and sort of philosophical type questions that I'd like to ask. I guess the first thing that strikes me about this is the seeming inevitability of calcification. And this idea that a smooth muscle cell just somehow inevitably goes down this osteoblast lineage. I'd like you to sort of expand a little more on what you think about that. And for example, you showed the Waddington landscape, that sort of classic picture. Do you think of an osteoblast as being some lower energy state than a smooth muscle cell? It's just eventually going to collapse into that state? I mean, why the inevitability of this process?
Highly esteemed opponent, thank you very much for being present here today, as well as for your compliments. With regards to the inevitability of calcification, actually, I would like to say here that I don't think there is an inevitability that calcification will occur. There is a possibility that some of us can develop vascular calcification, depending on our lifestyle and how long we get to live as well. So what I propose is that there is this inevitability of this smooth muscle cell, of this subpopulation of smooth muscle cell that will calcify. Coming back to the Waddington landscape, what I hypothesize is that actually the smooth muscle cell, being the plastic cell that it is I don't think that every cell in our body comes to the bottom of the hill. It can't be. That we have somatic stem cells in our body keeping us alive is part of this. And I wonder if the smooth muscle cell actually, as an entity with the way in which the cardiovascular system presents itself first during development, doesn't make its way all the way down the hill and is somewhere up on the ledge waiting to fall down, if you will, or to stay up if we maintain a good lifestyle. I think that's evident by the plasticity which the smooth muscle cell has, as well as by the timing of looking at cardiovascular development, and looking at how the bone systems, looking at how the skeletal system develops. It's a lot of the skeletal structures are mesodome derived, which occur via which develop via a microvascularization, which eventually loses calcification inhibitors and then allows mineralization of the vasculature to occur to form our bones. So I think there's a good few things to consider and to think about when looking at this. Yeah.
More of a technical question then. With the 3 types of smooth muscle cell you've generated, and you had that nice diagram showing how the lateral mesoderm and the various types map to the aortic arch. how accurately do you think your cultured cells are actually mapping to those regions? And if you look at calcification, is it that much stronger right at the beginning where the lateral mesoderm cells are? And does it sort of titer off as you go around the arch? And does that correlate well with your in vitro studies?
No, thank you for that question. So when we look at the clinic, and when we see cardiovascular calcification really presenting itself with regards to aortic calcification, in the majority of cardiovascular disease patients, we see that it starts at the root of the aorta, at where the valves are. And so depending on the stage of calcification development, you have calcification present at the root, and then maybe in patients who have more progressed calcification, it really extends out into the arch. So this is where I think that the findings presented in this thesis present that, whereas traditionally people expect this to be due to biomechanical forces and differential stress and strain, that potentially there is a smooth muscle cell subpriming.
Right. So you talk about it as a temporal thing, like it starts at the root and then spreads up, suggesting that eventually there's going to be equal calcification all around. That's not what your in vitro data show, where your curves turn over and the level of calcification in the NC and PMs is about half of what it is with the LMs. Is that what you see in the aorta? Do they never get to that level of calcification that the root does?
Yeah, thank you for that question. So with regards to the model with the assay, so when I see the curving off, if you will, that's when when I'm looking at my data again and being a bit like self-critical, I wonder if maybe I should have more probe in there. If maybe just is kind of like the probe has kind of maxed out, if you will, or you know, I add equal sums of probe to each condition, but if maybe at this point in time the probe in solution has dissolved or is no longer present and should have been refilled. So that's what I would say. That's what I kind of wonder when looking at the flattening off, as well as with regards to it being.
Well, but your PM and your NC flatten out well below where your LMs do. So that's the flattening out may be an artifact of substrate loss, but still they flatten out half of what the others do. Anyway, that's a technical question. With regard to the single-cell-seq that you did, you note that the LMs, which are more prone to calcification, express BMP2 and 4, which was interesting. Those are the ligands. Did you see their canonical receptors? Were they expressed, suggesting there was an autocrine loop there?
Pardon me? Can you?
Did you see the receptors for those ligands also expressed?
In terms of differentially expressed,
on the LMSMCs, you showed in your single-cell-seq that they're expressing BMP2 and 4. I just wondered, are the receptors present, suggesting there could be an autocrine driving of this osteogenic phenotype?
Yeah, so...
Please give a short answer. Please give a short answer.
No, but we could look better. Because this is purely a go term on regulation of ossification, and I haven't had the chance to go back and check whether or not BMP receptors are within this go term.
Yeah, I think there's a lot still in your single-seq data that you can extract. Yeah. Thank you.
Thank you, Professor Hughes. The next opponent is Professor Willy Janendiscent. He is Professor of Biointerface Science at RWTH Aachen University. Professor Janendiscent.
Dear candidate, first of all, like everybody, I'd like to commend you on this fine piece of work, especially on the drawings, which I like very much. And I would like to start with a question on the drawing on page 11. That's this dripping shower. That's an aortic branch, I think, an arch. And I don't know how to put this. To a cardiologist, everything has to do with the heart. To a hepatologist, everything has to do with the liver. And to an embryologist, everything has to do with the embryological origin of whatever tissue is affected. However, these vessels have more than smooth muscle cells. Can you tell us where the rest of the cells comes from? And if the origin is maintained and the properties of the origin are maintained in transplantation experiments, for instance, are you aware of any?
Highly esteemed opponent, thank you very much for being present here, as well as thank you very much for the years in which you were Aachen representative of the IntraCare Consortium, for which I was fortunate enough to be a part of, which funded this PhD work. With regards to to what other cell types are present within the vasculature and their developmental origin, so we have endothelial cells of the innermost cell type of that, of the vascular system, and these arise via various waves of development. So you have first an embryonic endothelium, which is derived from when you have your lateral mesoderm. Then lateral mesoderm goes in towards a hemioangiogenic cell type, and then this forms the endothelial cells which have primary angiogenesis. You then have this secondary wave of endothelialization within development. And as we stand here today, our endothelium is actually under a relatively quicker turnover, if you will, that's maintained by circulatory cells as well as possibly residential stem cells. Further, so that's going away from the smooth muscle to the inner layer. Within the outer layer, we also have cell types which are referred to by some as fibroblasts, as well as possibly adventitial stem cells. I believe also lymphocytes are present, and nervous tissue can be present as well within the adventitia, as well as a large degree of extracellular matrix.
And will they all be of the same embryological or of the same tissue origin, depending on the position along the aorta? So how I understand the lineage tracing work of the Majeski group, so when we're looking at, let's say, the root of the aorta, what seems to happen is you have this ball of like cardiac development, and then the neural crest really comes in and overlaps around it, and so there's this suggestion that the neural crest is maybe more on the outer aspects of the ascending aorta, and the lateral mesoderm is on the middle. But this is from mouse lineage tracing model, so I don't know how relevant that would be within the human situation. So it may well be that the functioning tissue actually is a mixture of cells from all places in the body.
I mean, for sure, that's what happens. I don't believe that there is a hard line within the aorta. Yeah, that says that, okay, here is where the lateral mesoderm will stop and the neural crest, there is overlap, and we know within the developing cardiac system, the neural crest plays an important role on that. So actually individuals who are unfortunate to have what we consider to be pathological mutations to the neural crest experience difficulties such as improper valve formation.
And when I look at the picture, I see this bend, and I see the blood flowing through it. So for physiologists, there is a large influence of the biomechanics of the flowing blood, or of the blood flow and calcification. And there's predilection sites that every clinician knows. For instance, there's rooms behind the heart valves that are less well ventilated, if you so will, than others. And there's areas of high flow and low flow. Can you maybe explain what that may have to do with calcification? Because it doesn't primarily affect the smooth muscle cells.
Yeah, that's a good point. With regards to the differential flow, this, by having a higher degree of flow and let's say stress on a particular branch of the wall, increases the need for turnover and for maintenance of homeostasis. And I think that while you have this primary knock to the endothelium, should this be exasperated further and further throughout, quite continuously, this will go on to affect the whole tube. If you keep hitting on the inside of a tube, okay, maybe you have the damage initially at the inside, but it will reach through. And so while I don't wish to say that biomechanics has no role within calcification, I think it has a very important role within the development of aortic root calcification. But I think the smooth muscle cell subpopulation and diversity of them is one of maybe the influencing factors with this differential mechanical force.
Okay, my last question is, You probably know by now that I'm not a big believer in the calcification equals bone formation theory. Actually, I'm totally opposed to it, especially in the in vitro work and in cell culture work, where we have very, very high artifacts stressing cells, and maybe that drives them into calcification. Everything we study is just paraphernalia. In real life, calcification and bone formation are associated in very, very late stages. But most of what we consider vascular calcification has nothing to do with bone formation. There's not a single bone cell around. So I wonder if you had a look at the stress response in your cells, or at age-related expression patterns, or even at what Kathy Shannon already mentioned, at transitory apoptosis or cell death signatures. Can you comment on that? Because we just stress the cells, and if they don't survive that, they die, and then they calcify.
Yeah, so this is work in progress. If you were to look at to further delineate kind of like what the mechanisms are, but I think one thing which we can appreciate from the work which I present here is that the lateral mesoderm doesn't undergo bone formation itself. It's just that you have this coincidence of genes present which cause bone formation.
Mr. Akbulut, you may have noticed that the time appointed for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and the quality of your defense. And please await our return with the results of our deliberation.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Mr. Akbulut, the degree committee has assessed the quality of your thesis and the quality of your defense. And in view of this positive verdict, and of course in taking into account the your previous qualifications, the committee has decided to grant you the degree of doctor. And Professor Schurgers is authorized to confer upon you this academic distinction in accordance with Dutch University custom and law. And I invite your supervisor now to take the floor. Professor Schurgers.
So before we start, Cengiz, the first words. Do you promise to work in accordance with the principle of scientific integrity at all time, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Asim Cengiz Akbulut, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Whole book of papers. Dear Dr. Akbulut, dear Cengiz, finally, the well-deserved title is in the pocket. I'm the first one to congratulate you on this very great achievement. Looking back 5 years, I can honestly say, time flies if you're having fun. It was a great pleasure to take this ride with you together. And in the next few minutes, I will try to refer to some of our moments, philosophical ones, in the past. But first, I want to congratulate also your supervisors from Cambridge, Professor Sanjay Sinha and Professor Rafael Kamon from Aachen, to do this together. I think we had a great time and it was a great achievement. Thank you for supporting this important project. And I hope this is not the end of our collaboration, or not even the beginning of the end, but it is perhaps only the end of this beginning. Also, I want to extend my congratulations to your family and to your friends. And also, of course, to Selene. Hi, Dr. Prisco. Next, I want to thank the members of your thesis assessment committee present here today in person, which is great after a time of 3 years with COVID, for their involvement and participation. Professor Hacking, as coordinator of IntriCare, but now also as chair of this committee. Professor Baker from Maastricht and Edinburgh, as we heard. Professor Schneider from Aachen. Professor Jan en Deggen from Aachen. You were even present in my PhD thesis defense more than 20 years ago. Thank you for again being here. Professor Shanahan, thank you for taking the trip from London all to Maastricht, and of course Professor Hughes. But you're also a little bit Maastricht now. So dear Cengiz, our first meeting dates back to the mid-2017, and it all started with the Horizon 2020 IntriCare, in which we wanted to investigate the role of microcalcification in vascular remodeling. 15 ESRs, early stage researchers, to be selected from all over the world. The Netherlands, Belgium, Germany, Greece, Poland, Romania, Russia, Brazil, Pakistan, India, China, and finally, but not least, England. A wonderful group of young, talented researchers that applied for a training in a joint and double doctorate training program. For the position of ESR3 in IntriCare, we had some 25 candidates applying for the position. A very popular job description, as it turned out. We selected 3 candidates, invited them for an interview in Maastricht. I could not describe it better than how you phrased it yourself in your acknowledgement of your thesis. A blundering buffoon in the wrong suit. Believe me, Cengiz, this impression is burned in my brain and likely stays there forever. So thank you for that. I remember you were very nervous during that presentation, wanting to make a very great impression. However, it must be said, you stood up, you gave a presentation that was spot on. Not taking the easy way, talking about a bachelor's or master's thesis as the others did, but on the topic of ESR3 itself, depicting how you would see the outline of this project. And it was clear that you were, Cengiz, a diamond in the rough. That Cengiz was and is a special person became clear from the beginning, maybe because of his Turkish roots combined with some British influences. A famous Turkish saying by Atatürk is, "Our true mentor in life is science." And an English one by Winston Churchill is, "Success is not final. Failure is not fatal. It's the courage to continue that counts." And I think these phrases perfectly fit your person. Within the IntraCare training program, we want to deliver the next generation scientists with emphasis on science, technical and clinical and valorization skills, as well as communication and collaboration skills. And to reach these goals, it was mandatory to have a very detailed training plan. However, with Cengiz, nothing happened or happens as originally planned. His project, working with primary vascular smooth muscle cells and investigating the phenotypic switching, was already in the first weeks of the PhD challenged and moving towards an iPSC generation. Cengiz tried to convince me of the importance of this work, but to me, this was deviating from the plan. And somewhat new. Was I risking this great project and follow this young, eager student? It was Atatürk who phrased, "The biggest battle is the war against ignorance." And to some level, I was very ignorant to this new world of vascular stem cells. In line with this, I want to refer to a paper that was published in 2008 by Martin Schwartz in the Journal of Cell Science, with the title, "The Importance of Stupidity in Scientific Research." In this paper, it says, "We just don't know what we are doing. We can't be sure whether we are asking the right question or doing the right experiment." And this phrase assured me a little bit. The paper continued, "Productive stupidity means being ignorant by choice. Focusing on important questions puts us in the awkward position of being ignorant. One of the beautiful things about science is that it allows us to bumble along, getting it wrong time after time, and feel perfectly fine as long as we learn something each time." With this in mind, I thought, what could go wrong? And thinking, I don't do a good job of teaching PhD students how to be productively stupid. That is, if we don't feel stupid, it means we are not trying really. So, stem cells it was. Cengiz wanted to do his secondment, another requirement of the European ITN grant, in stem cell research. And also here, Cengiz did not follow the strict guidelines set out by the EU. So we had to change the original plan for a 6-month visit to Cambridge. It was worth mentioning, or it is worth mentioning, that Cengiz applied for and received the prestigious HS BAFTA grant for this. So it was done. The secondment was changed to the Cambridge Stem Cell Institute under the guidance of Professor Sanjay Sinha. Here, Cengiz learned about iPSC differentiation towards vascular smooth muscle cells of different embryonic origin. This was just before COVID-19, capturing the world and isolating us for almost 2 years. But you brought back, just in time, important knowledge. And that technology, and together we were able to build a future around that. Not only for our own group, but also for biochemistry, for CARM, and even for the whole FHML. Most people here present today are aware that there is now, as of this year, a stem cell facility in Maastricht, to which you, Cengiz, contributed significantly. You were the first employee of this facility, and now in your role as lab manager, further shaping this future of cardiovascular stem cell work in Maastricht. About this scientific knowledge, we had no doubt. Next, it was to train Cengiz how to grow in his position. And so Professor Hacking and I, we invited Cengiz to join us to Irvine for our collaboration with the University of California, Irvine, with the group of Professor Hughes. Here we could work on his communication skills as well as acting in such setting. I can say, Cengiz had to learn some lessons, and it must be said, he listens and takes this serious. At our last day before flying home, we went to Borrega Spring for a great hike, and Cengiz took also this role very serious, carrying a few beers and ice in his rucksack. We still thank you for that, Cengiz. It were one of the best and finest cold beers. I'm sure, Cengiz, that your future is bright in Maastricht because you also love Maastricht. In the summer of 2019, you bought your own house in Maastricht. And since recently, you even managed to get your driver's license in Maastricht. And now you drive yourself to Aachen for our scientific collaborations. You also found your hobbies in Maastricht, music and dancing. I first noticed this skill when celebrating my inauguration with a party. It was you that was first on the dance floor and forcing many others to follow. 5 years ago, I was not even aware of a Lindy Hop Society in Maastricht, named after the famous aviator Charles Lindbergh, nicknamed Lucky Lindy, who hopped the Atlantic. This dance, you young grasshopper, fits you perfectly. During one of our great biochemistry days out, you were in the organization, and you thought it would be fun to see some hardcore scientists dance. I can tell you, forcing the chair and vice chair of biochemistry on the dance floor nearly costed you your PhD position. Cengiz is creative in many more ways, also in music. It was on these occasions that I was attending a conference and that our group meeting was held by the PhD students themselves. It was during that meeting I received a video via WhatsApp that our vascular aspects group had suddenly an anthem. It turned out to be the microcalcification. And of course, this was written and played by Cengiz. And not in the last place, his creative art impressions, as can be acknowledged during his presentation today, but also in his thesis. Schematic representations of stem cell work, and a great pleasure that you always include some vitamin K in that as well. You even work now on MGP, my favorite protein, for over 25 years. Some advice at the end. A great person once said, courage is what it is to stand up and speak. For sure, Cengiz, this is what you master. Courage is also what it takes to sit down and listen. In your new position as Dr. Akbulut and manager of the lab, you will need to challenge this. I'm sure that you will, in time, master this as you are eager and very fast learning. I will end this laudatio with music. A great song by Prince, one of your favorites, and it's called Sometimes It Snows in April. In this song, he poetically sings that all good things, they say, never last, and love isn't love until it's past. For our collaboration, Cengiz, and our friendship, I hope this is not true, and that we may work together for many more years, that we may have our philosophical discussions about science, politics, religion, music, or art. With that, I now give the word back to the prorector.
Esteemed Dr. Akbulut, dear Cengiz, it's my great pleasure to congratulate you with your doctorate and the honor that you have acquired today. And I do that also on behalf of the Board of Deans of Maastricht University. I would like to share some impressions of the committee with you. We have seen a very original and fantastic presentation. We call it in Dutch lekenpraatje, for the lay people. Very clear. We enjoyed it. And we have seen a good, well-written, a sort of monographical thesis. And that was focused on a difficult and complex subject. And you have used in your studies good techniques. And there came out really interesting issues. And that was very much appreciated by the committee. And in your defense, we saw a very good defense. And we enjoyed your, let me say, your special style, which sometimes circumvented the questions, and in general were very broad and general, superficial, and sometimes we would have liked some little bit more depth, but we have really seen a real PhD, someone who is really a doctor of philosophy. And I congratulate you with your thesis, with your defense, and the result of your studies, and I think you will have a wonderful professional career. And I would like to congratulate your 3 supervisors with this result that we have seen. The thesis, the studies, and the defense. You may be proud of this result. Congratulations. And of course, I'm not finished yet. I would like to include in my congratulations your family, your parents, other members of your family. Congratulations with this wonderful result. You may be proud. And I also include other members of your family, so not only the people that are sitting here on the first row, but also other family members, and of course your friends and colleagues and all those who have contributed to the results of your studies in the form of this thesis. I would like to thank also the members, all the members of the assessment committee as well as this degree committee. Thank you very much. Almost all of you were also external guests, and Maastricht University appreciates your contributions. Thank you so much. And before I close this academic ceremony, I would like to ask you here on the first row, the near family, to stay here and walk with us to the hall to take a picture from the stairs. That's what we do regularly. And I would like to ask the other people in the aula to follow the beadle to the reception, which is in the rafter of this building. And you will find it if you follow the beadle, and we will come a little bit later. Hereby, I close this academic ceremony.